Welcome to SR Pharma again. So today we are going to discuss on the action potential in neurons. Mainly we are going to focus on the introduction of action potential and generation of action potential in neuron. Now coming to the action potential. Now in our first class, we have discussed about the resting membrane potential and electrical signal. Already we have discussed that there is electrical signal which is generated by the nerve cell are mainly two types. One is the graded potential and another is action potential. Now, nerve cell generates the signal that transmit the information. In our last class, we have discussed about the difference between the graded potential and action potential. We have seen that graded potential serve as a short distance signal. It is the short distance signal, whereas action potential or nerve impulse, it is also called as a nerve impulse, which signal over long distance. Now action potential are considered as a fundamental units of communication between the neurons. It is a fundamental unit because it is responsible for the transmission of signal over the long distances, one neuron to another neuron. Action potential is a quick rise and subsequent fall in membrane potential. So if we see about the action potential, we can see that quick rise, that means depolarization, and subsequent fall, that is the repolarization, that means the changes in membrane potential. As in our first class, we have details discussed about the membrane potential and resting membrane potential, and these changes are in a characteristic pattern. Why we are talking about that? Because first is there is a depolarization followed by the repolarization. We have told that action potential in neurons are also known as a nerve impulse or spike. Now if we consider about this depolarization, it is mainly due to the opening of voltage gated sodium channel, and repolarization, it is mainly due to the opening of voltage gated potassium channel, opening of voltage gated potassium channel, that is for the repolarization. So today we are going to discuss about the generation of action potential in detail. Now whenever we are talking about the generation of action potential, first important point is membrane potential. Now in case of membrane potential, we know that resting membrane potential in neuron is minus 70 millivolt. That means if we are considering the cell, for example, if we are considering this is a cell, now inside the charge, it will be minus 70 millivolt. That means 70 millivolt less compared to the extracellular side. That means in the cytosol membrane potential, it is 70 millivolt less. Okay. Now we know that 70 millivolt is the resting membrane potential. Resting membrane potential. Now this stimulus, it is considered as a sub-threshold stimulus. Sub-threshold stimulus. In our last class, whenever we have discussed about the graded potential, we have told that this type of the stimulus is not sufficient to produce the action potential. It is not sufficient to produce the action potential. So that is known as a sub-threshold stimulus. Next is, this is the called the stimulus. This is the stimulus which goes up to the threshold point. Goes up to the threshold point. So this is the threshold point, which is minus 55 millivolt. Minus 55 millivolt threshold point. So when the stimulus, if there is a stimulus, and due to this stimulus, if the membrane potential goes to minus 55 millivolt, then what will happen? It will cause the opening of voltage gated sodium channel. It is caused the opening of voltage gated sodium channel. As a result, membrane potential is increasing. Membrane potential, if you can see that membrane potential is increasing, which is known as a depolarization. Which is known as a depolarization. That means if we are considering this is a cell, so we know that in the extracellular side, sodium concentration is more than the inside. So whenever the voltage gated sodium channel is open, sodium, it will come inside. Sodium is coming inside. Now sodium is positively charged ion. Positively charged ion. So sodium is coming inside. Therefore there is a polarization shift. That means membrane potential is increasing. Membrane potential is increasing, which is known as a depolarization, and it is goes up to plus 30 millivolt. It will go up to plus 30 millivolt. That means in this particular point, if we are considering this point, inside it is the plus 30 millivolt. Inside it is the plus 30 millivolt. That means inside it is more compared to the extracellular fluid. In this point, when the membrane potential about to reach plus 30 millivolt, then there is a starting of closing voltage gated sodium channel. Starting of closing voltage gated sodium channel. So membrane potential, it is not crossing plus 30 millivolt. Plus 30 millivolt. In this condition, what is happening? There is a opening of voltage gated potassium channel. Opening of voltage gated potassium channel. Now if we are considering this, generally potassium concentration is more inside and less in outside. So as soon as the voltage gated potassium channel, it will be open, the potassium, it will go outside. Potassium, it will go outside. Now positively charged ion, it is going outside. Positively charged ion is going outside. Therefore membrane potential is decreasing. Membrane potential is decreasing, which is known as a repolarization. So it is membrane potential coming back to the resting membrane potential. So this particular phase, it is known as a repolarization. It is known as a repolarization. And in this stage, there is a starting of closing voltage gated potassium channel. There is a starting of closing voltage gated potassium channel. And if we are talking about the sodium channel, of course in this area, this sodium channel, it will be get closed. It will be get closed. So we can say that action potential can be started only in the area. If we are considering a neuron, then we can say that only in the area, that is the axon hillock of the membrane where the voltage gated sodium channel is abundant, because it is required that opening of voltage gated sodium channel, voltage gated sodium channel. So here we can find that there is a starting of closing the voltage gated potassium channel during the repolarization. This particular phase, it is known as a hyperpolarization. It is known as a hyperpolarization. Now, what is meant by the hyperpolarization? If still here also, if you consider here also, still the voltage gated potassium channel is still open, or in the other case, if the resting membrane potential goes down, consider to resting membrane potential, that is minus 70 millivolt, then it is known as the hyperpolarization. And again, in this condition, like sodium potassium ATPase, it will again it will going to activate, and then what will happen, the nerve, it will come back to that particular area, come back to the resting membrane potential. It will come back to the resting membrane potential, so that next action potential can be generated. So this is about the generation of action potential. In a nutshell, if we are going to discuss, when a stimulus it is coming, and when the stimulus goes to minus 55 millivolt, that means due to the stimulus, if the membrane potential goes to minus 55 millivolt, which is a threshold point, then what will happen? It will cause the depolarization, because due to the opening of voltage gated sodium channel, and membrane potential, it will rise up to plus 30 millivolt. So in this area, there is a starting of closing of voltage gated sodium channel, and into the peak there is a opening of voltage gated potassium channel. So as soon as the potassium channel it will open, and there is a, for example in this side there is a closing of the voltage gated sodium channel, so there is a repolarization phase. Slowly membrane potential it will come down, it will goes to the resting membrane potential. Slowly again it will attain the resting membrane potential. So this is the generation of action potential. Now, in general, in average, in a neuron, action potential lasts for only 1 millisecond, 0.001 second, 1 millisecond. That's why we are talking that action potential, it is brief, rapid, and large change into the membrane potential. Brief, rapid, and large change into the action potential, because there is a, it is going from the minus 70 millivolt to plus 30 millivolt, so almost the 100 millivolt change. So this is about the action potential. Now we'll see that during the generation of action potential, what is the changes in sodium channel. So in the resting membrane potential stage, when there is a resting membrane potential, sodium channel are in the resting stage or non-conducting stage, resting or non-conducting stage. Sodium channel, they having the two gates, one is the activation gate, another is the inactivation gate, activation and inactivation gate. So in the resting stage, activation gate is closed but inactivation gate is open. But sodium cannot come inside because activation gate is closed. So sodium cannot come inside. Due to the stimulus, when the membrane potential reaches to the threshold point, that is the minus 55 millivolt, this will trigger the opening of voltage gated sodium channel, So this stage it is known as the activated stage or conducting stage, activated or conducting. In this stage, both the activation and inactivation gate is open. So sodium, it will coming inside the cytosol. It is coming inside the cytosol and membrane potential rises. Membrane potential rises, which is also known as a depolarization. Depolarization. And during repolarization, we can find that this sodium channel are in inactivated stage, inactivated or non-conducting stage, because though activation gate is open but inactivation gate is closed. Inactivation gate is closed, so again sodium cannot come inside. Again the sodium cannot come inside. So this is the three stages of sodium channel. This is the three stages of the sodium channel. And for potassium channel, if we are going to discuss, during the resting membrane potential and depolarization stage, the potassium, voltage gated potassium channel, it will be voltage gated potassium channel, it will be closed during the resting membrane potential and depolarizing phase. But during the repolarization, the potassium channel, clear, potassium channels are open. Potassium channels are open. So this is about the sodium channel and potassium channel during the generation of action potential. Now coming to the refractory period. What is mean by the refractory period? So this is the period of time after an action potential begins, during which an excitable cell cannot generate another action potential. Now what does it mean? For example, if we are considering from here to here, this area, already in this stage, due to the stimulus, one action potential it is started, and in this stage again it is come back into the resting membrane potential. So that means refractory period is the stage in which after starting of generating one action potential, another stimulus cannot able to generate another action potential. That means it will come back, action potential, it will come back into the resting membrane potential, and again from here another action potential it will be generating. Now generally refractory period are two type, or we can say that refractory period we can classify into two stage. One is the absolute refractory period, and another is the relative refractory period. Absolute and relative. Now what is the difference? Now this stage it is known as a relative refractory period. What does it mean by the relative refractory period? This is the period of time. Clear? This is the period of time, as we have discussed about the time here. It is the period of time during which a second action potential can be initiated, during which means when it is near to the resting membrane potential, another action potential can be initiated, but only by a larger than normal stimulus. That means if very strong stimulus it is coming, then only another action potential can be generated. In normal stimulus, another action potential it will won't be generated, but if it is very strong stimulus, then there is a chance that another action potential may be generates. And coming to the absolute refractory period, in this stage, clear, in this stage or in this time period, even a very strong stimulus cannot generate second action potential. So during this period, a very strong stimulus also cannot able to generate another action potential. So mainly if you see that in this period there is a activation and inactivation of the sodium channel, we can see that. Now here already in the last slide we have discussed about the inactivation of the sodium channel. If we go back to the previous slide, we can see that resting stage, activating stage and inactivating stage. That means from the inactivating stage, a sodium channel cannot open. It should go to the resting stage. After that it should go to the resting stage, then only it will go to the activation stage. That means from the inactivation gate to activating stage, it is not possible. It must go to the inactivated stage to resting stage and then again the activation gate. That's why we are telling that in this period another generation of another action potential is not possible. While in case of the relative refractory period, very strong stimulus can generate the another action potential. So this is all about the refractory period. Now again in a nutshell if we are discussing, so this stage, this is the first resting stage. So it is the sodium ion. So sodium ion more into the extracellular fluid, and B is the potassium ion. So potassium ion more into the cytosol. So this is the resting condition. Next is depolarization. This is the depolarization. This stage, depolarization. In this stage, what is happening? If you see that here, this is the sodium channel plus sodium channel. So sodium channel is open. But potassium channel and pump, sodium potassium pump is closed during the depolarizing stage. Next is repolarization stage. This stage, repolarization stage, sodium channel is closed, but potassium channel is open, and also sodium potassium pump is closed. And during the refractory period, last stage of the refractory period means here we are telling that last part of the refractory period, when this is the time actually during the hyperpolarization, and then it is goes to the resting membrane potential, hyperpolarization, and then it will attain the resting membrane potential. That stage, sodium channel is closed, potassium channel is closed, but sodium potassium pump is open, and through this pump potassium is coming inside, sodium is going outside, and again it is maintain the resting membrane potential. So this is all about the generation of action potential. And if we see about a neurons, because we are talking about the action potential which is transmitted through the neuron, so we can find that this is the input zone. Through the input zone, that means receives the incoming signal from the other neuron. This is the trigger zone, axon hillock or trigger zone. That means here the action potential it will be initiating. This full area is a conducting zone. Because the action potential is conducting, it is propagating through the neuron. And this is the output zone. These are the output zone that due to the signal, neurotransmitter is releasing or they are sending the signal to the next neuron, like that. So that is the output zone. So this is all about the generation of action potential. So hope you have understood it in details. We have discussed. In our next class we will discuss about the propagation of action potential. Propagation of action potential. So this is all about the attribution and references. So thank you for supporting us, and hope this classes it will be helpful to understand different topics related to the pharmacology and pharmaceutical chemistry. Thank you once again.